Welcome to the Deep Dive. We're here to cut through the noise, get you well informed on topics that, well, really matter. Today we're plunging into something maybe you haven't heard a lot about. But it's this kind of hidden global threat. Doesn't hit humans directly, but it can unleash just widespread devastation, chaos even, affecting what we eat, the livelihoods of millions. We're talking about African Swine Fever, ASF. It's a classic example of this kind of silent but, wow, incredibly potent force.
That's absolutely right. African swine fever, ASF, we call it devastating transboundary animal disease. And the implications are profound for animal health, sure, but also global food security, international trade. It touches everything. And it's crucial, as you said, to understand it's not zoonotic. It doesn't jump to humans, but its capacity to just wipe out pig populations, that's a crisis on a truly global scale. And, for so many years, the fact there wasn't a widely available effective vaccine meant our only real tools were biosecurity, trying to keep it out, and unfortunately culling, which leads to these catastrophic economic losses we've seen worldwide.
Right. And that's our mission today. For you listening in, we've waded through, stacks of comprehensive analyses, the latest research papers, expert reports, all on ASF. Our goal, to pull out the absolute must-know nuggets of knowledge, the key insights. You'll probably hear some surprising facts, get the critical context you need to really grasp this complex topic, quickly but thoroughly. So let's start at the beginning. What exactly is this virus, this ASFV, that's causing so much chaos?
Okay, so the African swine fever virus, ASFV, it's genuinely unique in the world of viruses. It stands completely alone. It's the only member of its viral family, the S. Forviridae, and here's a kicker. It's the only known DNA virus that's transmitted by arthropods, specifically soft ticks, the Ornithodorus genus.
Wow, a DNA virus spread by ticks. That's unusual.
It really is. And the virus itself, think of it like a microscopic tank. It's large, complex, multi-layered, structurally very intricate, about 175, 215 nanometers. Its genetic material, its genome is also massive, a big, linear, double-stranded DNA molecule. And it has these distinctive features at the end, sort of like covalently closed hairpin loops in repeated sequences. Very characteristic.
Okay, so it's big, complex, and I heard you say it's incredibly self-sufficient, almost carries its own toolkit.
Exactly. That's a great way to put it. Its huge genome, it codes for something like 150 to 167 proteins, over 150 distinct functions, gives it this amazing autonomy from the host cell it infects. Think about it. It barely needs to borrow the pig's cellular machinery. It replicates almost entirely out in the cytoplasm, setting up these things called viral factories. And it brings its own crucial enzymes, its own DNA polymerase for copying its DNA, its own transcription factors, even DNA repair enzymes. It's remarkably self-reliant.
Like building its own little factory right inside the pig cell. Does that self-reliance make it particularly hard to fight off?
It certainly contributes. And interestingly, a large chunk of that genome isn't actually for the basic replication, making copies of itself in a lab dish. It's dedicated to proteins that are absolutely critical for surviving inside the pig and causing disease. These proteins are masters of manipulation. They hijack host cell functions, and crucially, they help the virus evade the pig's immune responses. For instance, some proteins block apoptosis, that's programmed cell death, so the infected cell, the viral factory, stays alive longer. Others actively suppress the pig's immune alarm signals, things like interferons. And among all its components, certain structural proteins, we call them P72, P54, and P30, are really key. They're what we target for diagnostic tests, and they're prime candidates for vaccine development.
Okay, so it's complex, it's sneaky. How do scientists actually track its variations, keep tabs on how it's changing?
Well, the main tool we use globally is based on one of those proteins, the P72 gene. It acts like a barcode, letting us group the virus into at least 24 distinct genotypes. That's fundamental for molecular epidemiology, tracking where different strains are. And this is a really important point, especially for vaccines, the P72 genotype system can sometimes mask crucial underlying differences. The real action, the variation that affects how dangerous a strain is, often happens at the ends of the genome. There are these regions with multi-gene families, MGFs we call them, like MGF 360 or 505. Genes can be gained or lost here.
So two viruses could look the same based on that main P72 barcode.
Exactly. Same genotype, but they might have totally different sets of these MGF genes. And those genes play big roles in virulence, how sick the pig gets, and how the immune system sees the virus. This variation means two isolates of the same genotype can cause different clinical signs, different levels of severity, and trigger different immune responses. It's a major headache for developing vaccines that give broad protection against multiple strains.
Fascinating. And this virus, it has a deep history, right? Where did it come from originally?
It does. The thinking is it originated way back, circulating between ancestral ornithodoros ticks and wild African pigs, like warthogs and bush pigs. Now, in those wild hosts, it's usually asymptomatic. They carry the virus, the ticks spread it, but the animals don't typically get sick. It's a stable, ancient cycle. The modern forms, the ones causing disease in domestic pigs, seem to have emerged later, maybe around 1700 AD. That timing lines up pretty well with when domestic pigs were first introduced into southern Africa.
So it's got this huge arsenal of immune-evading proteins. What's the bottom line, the practical implication for controlling the disease?
The bottom line is that this toolkit allows the virus to effectively disarm the pig's primary immune defender cells called macrophages, letting it replicate to incredibly high levels. That's what leads to the severe hemorrhagic disease. But here's the crucial insight for control, especially vaccines. Many of these immune-modulating genes, the ones that help it hide or fight back against the host, they're actually not essential for the virus to simply make copies of itself. And that gives us a clearer pathway for rationally designing live attenuated vaccines, LAVs. By carefully deleting specific virulence genes, we aim to create a weakened virus, one that can't cause severe disease but can still stimulate a strong protective immune response. This link between understanding how it causes disease and figuring out how to weaken it for a vaccine, that's really the core of modern ASF vaccine research.
That makes a lot of sense. Okay, so this incredibly complex virus gets inside a pig. How does it actually unleash all that havoc? What's the attack plan?
Right, so entry is usually oral or nasal. Think direct contact between pigs, or eating contaminated feed, drinking contaminated water. Tick bites are also a route, especially in the original African cycle. Once it's in, the first stop is usually the tonsils and the nearby lymph nodes. That's where initial replication kicks off. But then it spreads really, really fast. It gets into the lymphatic system, into the bloodstream, leading to what we call viremia, lots of virus in the blood. And soon you find high viral levels pretty much everywhere, in all tissues. The incubation period varies, maybe 4 to 19 days typically, but with highly virulent strains, you can see signs of acute disease in just 3 or 4 days. It can be incredibly rapid.
And which cells is it really going after inside the pig?
It has what we call a pronounced tropism, a strong preference for monocytes and macrophages. These are absolutely critical immune cells, the body's first responders and cleanup crew. ASFV targets them directly. It also infects endothelial cells, the ones lining the blood vessels, which definitely contributes to the bleeding problems we see. And how it gets into those macrophages is quite complex, involving processes like macropinocytosis, essentially tricking the cell into engulfing it.
And those immune evasion strategies we talked about, how does it fight back against the pig's defenses once it's inside?
Oh, it actively works to shut down the host's early warning systems. It's particularly good at inhibiting type IR interferon's key antiviral signals. But maybe the most devastating tactic is its ability to induce widespread apoptosis that programmed cell death again, but this time in uninfected bystander cells, specifically T and B lymphocytes.
Wait, it kills immune cells that aren't even infected?
Exactly. These lymphocytes are crucial for the adaptive immune response, the targeted memory-building part of immunity. By triggering their mass suicide, ASFV causes severe depletion of lymphoid tissues, think lymph nodes, spleen, leading to profound immunosuppression. It effectively cripples a pig's ability to mount a specific effective defense against the virus. It's like it decapitates the immune system right at the start.
So hang on. If it's not primarily the virus directly killing the blood vessel cells, what is causing all that horrific internal bleeding?
This is such a critical point to understand about ASF pathogenesis. The devastating hemorrhages you see in acute cases, they're largely not from direct viral damage to the endothelium. Instead, it's considered an immunopathological consequence. Basically, the pig's own immune response goes haywire. Infected macrophages pump out a massive, uncontrolled flood of inflammatory molecules, a cytokine storm.
Like the body's alarm system goes completely berserk.
Precisely. It's an overreaction. This cytokine storm causes blood vessels to become leaky, damages their lining extensively, and severely disrupts the whole blood clotting system. You get a crash in platelet, counts thrombocytopenia, and widespread uncontrolled clotting and bleeding, known as disseminated intravascular coagulation, or DIC. The end result is those extensive hemorrhages you see in multiple organs.
Okay, connecting this to the bigger picture then.
Yeah.
It's like a two-pronged attack. The virus multiplies like crazy.
Right, overwhelming numbers.
And it tricks the pig's own immune system into becoming the main driver of the damage.
Exactly right. It's a dual assault. The virus replicates massively, and it turns the host's defenses, particularly those infected macrophages releasing cytokines, into the primary agents of pathology, causing that systemic vascular collapse. And at the same time, remember, it's destroying the lymphocytes needed for a targeted counterattack. This explains why the disease progresses so rapidly and severely. It also tells us why any successful vaccine needs to work fast and potently to get ahead of this preemptive strike on the immune system.
Wow, okay. So after all that complex biology, what does this actually look like for the pigs themselves? When ASF hits a farm, what are the signs?
Well, it really varies. Depends on the virus strain, the pig's condition. But we generally talk about four main forms. There's peracute. This is sudden death, often with hardly any warning signs. Mortality can be basically 100%, just devastatingly fast. Then the most common is the acute form. Pigs get a really high fever, 40 to 42 degrees Celsius. That's like 104 to 107 Fahrenheit. They become extremely lethargic, refuse to eat. Mortality here is also typically near 100%, usually within, say, 4 to 20 days.
That's incredibly lethal. Are there less severe forms too?
Yes, thankfully. The subacute form is usually caused by less virulent strains. The illness is less intense, lasts longer, maybe 5 to 30 days. Mortality is lower, but still significant, maybe 30% to 70%. And finally, there's a chronic form. This comes from low virulent strains and can drag on for months. The signs are variable weight loss, maybe a low-grade fever that comes and goes, breathing problems, skin lesions that necrotize, swollen joints, arthritis. Mortality is low in this form, but the pigs are often carriers.
Okay, focusing on that calm acute form, what are the most striking things a farmer or vet would actually see?
Key things. That sudden high fever and the pig just being incredibly down, depressed. But visually, the skin changes are often the most dramatic. You can see this diffuse reddening erythema, which often progresses to a deep bluish-purple color that's cyanosis. It's most noticeable on the ears, the snout, the belly, and the legs. Signs of bleeding are also very common, maybe bloody discharge from the nose, bloody diarrhea. And you might see tiny pinpoint hemorrhages, petechiae, or larger bruise-like ones, ecchymoses, on the skin itself. Other signs you often see are vomiting, difficulty breathing, and in pregnant sows, abortions are frequent.
And if an animal dies, what are the telltale signs inside during a postmortem?
Right. The internal lesions in acute ASF are dominated by hemorrhages. But the most characteristic, the most consistent finding is the spleen. It becomes massively enlarged, very dark red or even black, soft, easily broken, friable. We call it hemorrhagic splenomegaly. Honestly, seeing that spleen is almost pathognomonic, meaning it's a sign strongly indicative of acute ASF. You also see lymph nodes throughout the body, especially in the gut mesentery, that are severely swollen and filled with blood, often looking like dark clots. The kidneys are another classic site. They often have these prominent pinpoint hemorrhages scattered across the outer surface, giving them what's often described as a perky egg appearance. Hemorrhages are common elsewhere, too. Heart, bladder, stomach, intestines. And you often find excess fluid, sometimes straw-colored, sometimes blood-tinged, in the chest and abdominal cavities.
This raises a really important practical question. With symptoms that severe, how do you know for sure it's ASF and not something else that might look quite similar?
That's exactly the problem in the field. The clinical signs and even many of the post-mortem lesions can look almost identical to classical swine fever, CSF, also known as hog cholera. You simply cannot reliably tell them apart just by looking. Laboratory confirmation is absolutely 100% essential, and there are other diseases to consider too in the differential diagnosis, things like bacterial septicemias, maybe swine erysipelas or salmonellosis, porcine reproductive and respiratory syndrome, PRRS, coursing dermatitis and nephropathy syndrome, PDNS. They can all cause severe illness and death.
So digging a bit deeper on that CSF comparison, what are the key differences a lab would look for?
Okay, so a few key distinctions. That massive hemorrhagic spleen is really characteristic of ASF. With CSF, the spleen is usually normal size, or it might have these small, dead areas along the edge called infarcts, but it's not typically huge and pulpy like an ASF. Another one. Chronic CSF can cause these things called button ulcers in the large intestine. You don't see those with ASF. And under the microscope, histopathology shows different things. ASF typically causes widespread destruction of lymphocytes and tissue death. CSF, on the other hand, often shows inflammation in the brain, but without pus formation, non-suppurative encephalitis. These details are critical for the lab diagnosis.
Okay, let's zoom out again. How did this disease, which was originally confined to parts of Africa, become this massive global crisis, hitting multiple continents? How did it escape?
Yeah, its global journey is really quite something. Understanding its spread involves looking at two distinct but linked transmission cycles. First, there's the original sylvatic cycle in Africa. This is the ancient stable cycle involving those asymptomatic wild soothes, warthogs, bush pigs, and the Ornithodorus soft ticks. In this cycle, the ticks are absolutely key. They're not just passive carriers, the virus replicates inside them. They act as a long-term reservoir, harboring the virus for years, and they can transmit it through generations of ticks vertically, horizontally, transstadially. They're like living viral banks.
Okay, so that's the wild cycle. What happens when it gets into domestic pigs or feral ones?
That kicks off the domestic feral cycle. Once ASFV is in these populations, transmission becomes highly efficient through routes that don't involve the ticks. Direct pig-to-pig contact is very effective. But a huge factor, especially for long-distance spread between regions or even continents, is the ingestion of contaminated pork products. Think uncooked or improperly treated kitchen scraps, swill feeding, or even contaminated feed ingredients, and fomites in inanimate objects. Trucks, equipment, boots, clothing. If they're contaminated, they can easily carry the virus from one place to another.
Now, you mentioned something really pivotal for Europe and Asia. This new kind of cycle emerged, the wild boar habitat cycle. How is that different?
This is a crucial shift. Unlike the African wild pigs that co-evolved with the virus and are largely resistant, Eurasian wild boar are highly susceptible. They get sick, they die from ASF, much like domestic pigs do, and their carcasses remain highly infectious in the environment for a long time, potentially months, depending on conditions. These carcasses act as persistent sources of virus. Combine that with the fact that wild boar are widespread, mobile, and sometimes interact with low biosecurity farms. It creates this incredibly challenging reservoir that makes eradication extremely difficult in Europe and parts of Asia. It's a wildlife management problem tangled up with the domestic pig problem.
Can you sort of trace the path? How did it jump from Africa and they explode globally?
Sure. So originally it was only in sub-Saharan Africa. The first big leap out was in 1957 into Portugal. That led to decades of it being stuck, basically, in the Iberian Peninsula, Spain and Portugal. Eradication there took a very long time. But the modern pandemic, the one causing the current global crisis, really kicked off in 2007. That's when the highly virulent genotype the second strain got introduced into Georgia, in the Caucasus region. From Georgia, it spread steadily through the Caucasus, into Russia, across Eastern Europe. It was a slow burn initially, but relentless.
And then China happened.
Exactly. August 2018. That was the catastrophic turning point. First detection in China, home to roughly half the world's pigs. That just ignited an unprecedented firestorm across Asia. It spread incredibly rapidly to Vietnam, the Philippines, South Korea, Indonesia, India. Over 20 countries in the region have been affected, and it's still active. Sri Lanka reported its first cases in late 2024. Then the Americas, which had been free for decades, got a major scare in July 2021. Confirmation in the Dominican Republic, followed quickly by Haiti. That put the entire Western Hemisphere on high alert. So as of early 2025, we're looking at reports from 83 countries across 5 continents just since 2005. Active outbreaks are ongoing right now in Africa, Europe, and Asia. It's a truly global problem.
So looking at the recent spread, what's the main engine? Is it still the ticks, the wild boar?
Well, the ticks are mainly relevant in the African context and parts of southern Europe. The wild boar are a huge issue in Europe and increasingly in Asia. But the primary driver for long-distance jumps, for getting into new countries and continents, it's unequivocally human activity. Transporting contaminated pork products legally or illegally is a massive risk. Contaminated feed ingredients, fomites carried on vehicles, equipment, even people's clothing or footwear moving between farms or countries. In Europe, yes, the wild boar are the main reservoir, but spillover into farms usually involves a biosecurity breach, contaminated feed, vehicles, people bringing it in. In Asia, the explosion was fueled by a combination of factors: complex, often informal, pig and pork trade networks, the very common practice of swill feeding, especially in backyard farms, and generally inadequate biosecurity on millions of smallholder operations. The ongoing outbreaks in 2024-2025 just confirm how entrenched it is.
So what's the fundamental shift we need to grasp about how ASF behaves now?
The key insight really is this transformation. ASF went from being this geographically limited, tick-dependent disease in Africa to a global pandemic driven mostly by human actions and its own incredible resilience in the environment and adaptability to new hosts. That 2007 introduction of genotype the second into Georgia and its subsequent establishment in Eurasian wild boar, that was absolutely pivotal. It created this persistent, hard-to-manage wildlife reservoir outside of Africa. So what this means is that effective control strategies have to focus heavily on managing human behavior trade, travel, farming practices, waste disposal, and crucially, managing wild boar populations where they are reservoirs. It's as much about people and wildlife as it is about pigs on farms now. And I'd reiterate, the establishment of ASFE in Eurasian wild boar is probably the single greatest barrier to getting rid of it regionally. Unlike the warthogs, these boar get sick, die, and their infectious carcasses persist. It's a massive environmental contamination problem.
Okay, shifting gears a bit. Beyond the devastating impact on the pigs themselves, what's the wider fallout? What does ASF mean for our global economy, our food systems?
The economic impact is just staggering. ASF is arguably the single most economically damaging disease to ever hit the global pig industry. We're talking cumulative losses easily in the hundreds of billions of US dollars, and the impact comes in waves. There are the direct losses, which are bad enough, the value of the pigs that die, which can be up to 100%, the costs of having to cull entire herds, sometimes preemptively, the expenses for diagnosis, surveillance, cleaning, disinfection. It all adds up incredibly fast. But often, the indirect losses are even bigger and last much longer. The moment ASF is confirmed, international borders slam shut for pork exports from that country or region. Trade restrictions, export bans, they're immediate. For a major exporting country, let's take the U.S. as a hypothetical example studies project, that just a single outbreak could lead to revenue losses anywhere from $15 billion to over $50 billion, mainly due to lost exports. It's economically crippling.
Can you give us a sense of scale with a real-world example?
Yeah.
Like China?
Absolutely. That 2018 outbreak in China is the starkest example. Remember, they have about half the world's pigs. Estimates suggest they lost around 225 million pigs due to death or culling. That's roughly half their entire national herd at the time. The direct economic losses from that event alone have been estimated as high as $141 billion. Just think about that number. In Vietnam, a smaller country but still a major producer, they culled nearly 6 million pigs in 2019, with estimated losses up to $4.4 billion. The scale is immense.
Wow. So it's not just losing the animals, it actually fundamentally reshapes the whole global pork market.
Completely. When a huge producer like China loses so much production, it creates this massive protein deficit that ripples outwards. Import demand surges from countries trying to fill the gap. Now, that might sound like an opportunity for ASF-free exporting countries, and it can be, but it also injects extreme price volatility into the global market. Prices can swing wildly, and the impact on food security is profound. Pork is a staple protein globally, over 35% of meat consumption worldwide. When ASF causes shortages, consumer prices for pork shoot up. This directly affects food accessibility for millions, especially lower-income households.
And I imagine this hits the smaller farmers, the backyard producers, especially hard.
Disproportionately so, particularly in Asia, where smallholder and backyard farming is very common. These producers often lack the savings, the insurance, the resources to implement high-level biosecurity or to recover financially after losing their animals. It can push entire rural communities, already vulnerable, deeper into poverty and food insecurity. It's a livelihood crisis for millions. So connecting the dots. An ASF outbreak isn't just about pigs. It sends shockwaves through the entire agri-food system.
Absolutely. It's a systemic shock. You lose millions of pigs. That creates a protein gap, driving up prices not just for pork, but also for alternatives like chicken as consumers switch. But then, fewer pigs mean less demand for animal feed. So that hits the markets for corn, soybeans, other feed ingredients. Trade flows get massively disrupted. Affected countries face export bans, sometimes for years. ASF-free countries face new risks, but also potential market opportunities. It reorders the entire global protein trade. And it's important to see the different kinds of economic damage. In places with lots of small farms, like Southeast Asia, it's primarily a crisis of rural livelihoods and local food supply. But in countries with highly industrialized, export-focused pork industries, like the US or Canada or parts of Europe, the most immediate and catastrophic financial damage would come from losing those export markets overnight. That would cause a domestic surplus, crashing prices for farmers, leading to severe financial distress. Two sides of the same coin, really. Okay. Given how incredibly devastating this disease is, economically and otherwise, what are the current battle plans? How do we diagnose it quickly and try to control it?
Well, first, as we said, lab confirmation is non-negotiable because it looks so much like other diseases, especially CSF. For diagnosis, the modern workhorses are PCR polymerase chain reaction and its more quantitative version, qPCR. These are the gold standards. They're fast, incredibly sensitive, very specific for detecting the virus's DNA. You can often detect the virus in samples like blood, saliva, or tissues even before the pig starts showing obvious signs of illness. That early detection is critical for trying to contain an outbreak. There are even advanced PCR techniques now for detecting carrier animals. Antigen ELISAs exist tests looking for viral proteins, but they're generally less sensitive than PCR, especially early on. Antibody ELISAs, however, are crucial. They detect the pig's immune response to the virus. These antibody tests are vital for surveillance, finding those chronically infected carriers, proving herds or regions are free of ASF for trade, and monitoring vaccine responses when they become available. Antibodies usually show up 7 to 10 days after infection and can last a long time. And things like testing oral fluid samples collected from ropes pigs chew on offer a really convenient way to screen whole groups of pigs.
But it sounds like there's a bit of a diagnostic challenge, depending on the stage of infection.
There is, absolutely. That's the fascinating diagnostic dilemma with ASF. If you have an acute outbreak with sick pigs, you need PCR for that rapid early detection before antibodies have even formed. But if you're looking for long-term carriers, maybe infected with less virulent strains or trying to confirm eradication after an outbreak, you really need serology, antibody tests. And complicating things, carriers might only shed the virus intermittently, making direct detection tricky. So you absolutely need a multi-pronged strategy. You need sensitive PCR for early detection in acute cases, combined with robust antibody testing for surveillance, finding carriers, and proving freedom. You can't rely on one. And because ASF is reportable to the World Organization for Animal Health, WOH, national surveillance programs are key. They usually combine passive surveillance that relies on farmers and vets noticing suspicious signs and reporting them quickly with active, targeted surveillance. That active part involves risk-based testing. For example, in the U.S., the USDA actively samples sick pigs submitted to labs, pigs condemned at slaughterhouses, feral swine populations, and pigs raised on farms that use garbage feeding, which is a high-risk practice if not done properly. International bodies like the FAO and WOAH help coordinate globally and provide early warnings.
So what does this all mean for how countries actually protect themselves, especially since there hasn't been a widely available vaccine to rely on? What are the core control measures?
Right. Without a universal vaccine shield, the foundation of ASF control rests heavily on what we call sanitary and phytosanitary measures. Basically, strict prevention and hygiene protocols. Biosecurity is number one. It is the single most critical line of defense for any farm or region trying to keep ASF out. This means having comprehensive plans. Key parts include strict access control, managing who and what comes onto the farm, having clear, clean, and dirty zones. Rigorous cleaning and disinfection protocols for buildings, vehicles, equipment, using disinfectants proven effective against ASFE, which is quite hardy, and a huge one, especially given how it's spread in Asia. A strict ban on feeding uncooked or improperly treated food waste, swill, or garbage feeding to pigs. That's a major pathway for introduction. Then, if an outbreak does occur, the response has to be swift and decisive. That usually involves immediate movement controls, a standstill order, stopping all movement of live pigs, semen, pork products within and out of the affected area. Control zones are set up around the infected premises, and unfortunately, culling is usually necessary for eradication. This means humanely euthanizing all pigs on the infected farm, and sometimes on neighboring farms too, depending on the risk assessment. Followed by safe and effective disposal of all carcasses and contaminated materials, bedding, feed, manure to eliminate the virus from the environment. It's a harsh but often necessary measure.
Okay, now for what feels like the million-dollar question, maybe even the billion-dollar question given the costs, vaccine development. This seems like the holy grail. Why has it been so incredibly difficult for, over 60 years?
It has been an incredibly tough nut to crack. The virus itself is just so complex, as we discussed. Its large genome, its sophisticated ways of interacting with and manipulating the pig's immune system. They've really thwarted traditional vaccine approaches. For decades, researchers tried making inactivated or killed virus vaccines. This works for many diseases. You kill the virus, inject it, and the body learns to recognize it. But with ASFV, they have consistently failed to provide good protection. The immune response just isn't strong or targeted enough. So the most promising avenue, and the one that's shown consistent effectiveness experimentally, has been live attenuated vaccines, LAVs. These use a weakened live version of the virus. The rationale comes from observation. Pigs that survive infection with naturally less virulent strains of ASF are often protected if they encounter a deadlier strain later. So the goal is to mimic that protective infection without causing severe disease.
Right. So what kinds of these live attenuated vaccines are being explored now? Are we getting closer?
We're definitely getting closer. There are two main types of LAVs being developed. Some are based on naturally occurring low-virulence isolates. Others, and this is where a lot of recent progress has been, are genetically engineered. Using modern molecular tools, scientists can now precisely delete specific genes known to be involved in virulence, those immune evasion or replication genes we talked about earlier. A really significant breakthrough came from the USDA Agricultural Research Service. They developed a gene-deleted LAV called ASFVGI177L. It's based on the pandemic genotype 211 strain, but with a key virulence gene removed. This candidate showed good safety and efficacy in trials against the deadly strain currently circulating globally. It's actually now licensed for use in Vietnam, and there are ongoing field trials in the Philippines and the Dominican Republic. That's a major step forward.
But I seem to recall you mentioning safety concerns with LAVs earlier. Is that still an issue?
Yes, that remains the persistent challenge with any live attenuated vaccine for ASF. There's always a background risk. Could the weakened virus still cause some level of illness, especially in vulnerable animals like young piglets or pregnant sows? That's residual virulence. Could it, over time, mutate and revert back to a more dangerous form? That's reversion to virulence. Could it establish chronic infections, leading to animals shedding the weakened virus into the environment? We have historical baggage here, too. There were disastrous field trials with early LAVs back in the 1960s in Spain and Portugal that actually caused widespread chronic disease. And even with the newer generation, those recent reports from Vietnam about potential reproductive problems in sows linked to a vaccine-like strain, they highlight that ensuring safety in large, diverse pig populations under real-world conditions is absolutely critical before widespread rollout. It's a major hurdle. Because of these safety concerns, there's also intense research into inherently safer platforms, like subunit and viral vectored vaccines. These don't use live ASFV at all. Subunit vaccines use just specific purified proteins from ASFV key antigens like P30, P54, or this protein called CD2V that are known to trigger an immune response. Viral vectored vaccines use a harmless carrier virus like adenovirus to deliver the genetic instructions for making those ASFV proteins inside the pig's cells. Historically, these safer platforms struggled to induce the strong, broad immunity needed for ASF, especially the crucial cellular immune responses. But there's been really exciting progress recently. I saw a study from early 2025 reporting a subunit vaccine based on that CD2V protein actually achieved sterile immunity in a challenge trial, meaning the vaccinated pigs were completely protected from infection. That's a huge landmark for a non-LAV ASF vaccine if it holds up. Another study showed a modified genotype 5 LAV could provide cross-protection against a different genetic group, genotype 3, although not against a much more distant one, genotype 9X. Still, it shows progress towards broader protection.
That sounds like real momentum. And I think I read something about new international standards for these vaccines.
Yes, that was another pivotal development in mid-2025. The World Organization for Animal Health, WOAH, officially adopted the first ever international standard for how ASF vaccines should be produced and evaluated. This is hugely important. It provides a globally agreed upon framework for assessing safety, efficacy, quality control, everything needed to ensure vaccines are reliable. WOAH is also explicitly warning countries against using poor quality or improperly tested vaccines and stressing that even with good vaccines, they're just one part of a bigger control strategy, not a silver bullet. Biosecurity remains essential.
So looking at this whole vaccine quest, what's the fundamental trade-off scientists are grappling with?
The bottom line in ASF vaccine development right now is this critical trade-off. You have the LAVs, which generally offer the best chance for strong, robust, potentially long-lasting immunity. They tend to work really well in inducing the right kind of immune response. But they come with those inherent safety questions. On the other side, you have the subunit and vectored approaches. They are much safer inherently, no risk of the vaccine virus itself causing disease or spreading, but historically it's been much harder to get them to induce protection that's strong enough and broad enough to reliably stop ASF infection and disease. Finding that sweet spot maximizing efficacy while guaranteeing safety, that remains the central challenge and the focus of intense global research effort.
So wrapping this up, what does this all mean? We've gone deep today into African swine fever, peeling back the layers from its unique, complex virus biology right through to its devastating global impact. Hopefully for you listening, you now have a much clearer, more comprehensive understanding of why this disease, even though it doesn't directly make people sick, poses such a significant and complicated threat to global food security, to economies, and to animal health worldwide.
Absolutely. And it's clear this crisis isn't just about, natural tick cycles anymore. It's really a reflection of our interconnected world, globalization, Modern farming, international trade, combined with this virus's incredible toughness, its environmental resilience, and the establishment of ASF in Eurasian wild boar populations, that's arguably the single biggest hurdle now for regional eradication. It's transformed ASF into this incredibly complex wildlife management and environmental issue, demanding unprecedented levels of international cooperation.
So as a listener, you've gained, hopefully, some crucial insights today into the intricate science behind ASFV, the profound economic and social shockwaves it sends out, and the cutting-edge work being done on surveillance, control, and crucially, the ongoing quest for effective vaccines. You now know why a virus most people might not have heard of can genuinely reshape aspects of our world.
And maybe a final thought, something provocative for you to mull over. Given the sheer genetic complexity of ASFV, its proven ability to adapt, to find new ways to spread, to establish itself in new hosts like the wild boar, what further adaptations might we anticipate from this virus in the future? And how will our global strategies need to keep evolving, perhaps even more rapidly, to stay ahead of such a dynamic, persistent, and formidable threat?